Hi, everyone. Welcome to my presentation in the frame of the Genetics Virtual Week by LabRoots. My name is Matthias Benoit. I am a research associate in Zach Lippman's group at Cold Spring Harbor Laboratory in New York, and I'm going to talk today about our recent research, and we're interested in the contribution of transposons in structural variation and phenotypic diversity in tomato. So my presentation today will follow two main parts. So in the first part of the talk, I will describe our most recent data about the study of the landscape of structural variation in tomato. And on the second part, I will provide a characterization of the transposon family that is involved in structural variation in tomato. So what do we mean when we speak about structural variation? Well, structural variation refers to large-scale structural differences that are found in organism genomes. And usually these variants are found as highly polymorphic within a species. So structural variants are made of deletions or insertion of novel DNA sequences. They can be due to tandem duplication, interstress duplication, as well as inversion and translocation. So such important rearrangement of the genomic DNA has the potential for really dramatic change in DNA organization. And it might have major impact on the biology of the genome and the way the genome functions. And indeed, structural variants have been associated with a lot of pathogenic conditions in humans, by example. So several cancers, as well as Alzheimer's or autism, have been associated with structural variation in a large number of cases. But structural variants are not only found in humans, and of course, you can also been associated with different process and behaviors in other species such as fish, in Drosophila, as well as bacteria or yeast. Structural variation is particularly important because it is associated with evolution of the genome, so also evolution of the behaviors of the organism. I'm presenting here a very interesting case of behavior change or evolution in the behavior of animals associated with structural variation. In this case, I'm showing that the adaptation of dogs to having the ability to rely on human food scraps to be eaten, and especially the ability of modern dogs to be able to process starch is actually linked to an increase in gene copy number of the genes that are able to produce the enzymes responsible for degradation of starch. And structural variation being this at the basis of the evolution of phenotypes, it's also particularly true in plants, where structural variation was actually instrumental during crop domestication. I'm showing here three different examples of how structural variation has actually enabled the identification, domestication, and selection of traits that were important for plant domestication and breeding. I'm showing here the fact that the loss of the shattering, which is the ability of the plant to disperse its own seeds for survival, so the loss of the shattering, which is a trait that is really important for modern agriculture, has actually been acquired through structural variation happening in genes that are involved in the shattering of the seed. So that was true in sorghum, but also in other plant species such as rice, where similar structural variance was involved in a loss of function of a gene involved in shattering. And another striking example is also the improved apical dominance seen in maize, which is due to the insertion of a transposable element upstream a gene that is involved in the regulation of the architecture of the plant. So altogether, they are compelling examples for structural variation being able to create new phenotypes and to provide a huge diversification in phenotypes within organisms. So in the lab, we are interested in plant crop, which is very important, being one of the most economically important plants in the world, which is tomato. And tomato as well shows a lot of traits that have been influenced by structural variation. I'm showing here 3 different cases in which the fruit features have been influenced by structural variation. The first example is the fruit shape that has been heavily influenced by structural variation. And here in this case, the elongation of the fruit has been linked to the occurrence of a 31 kb deletion in the promoter of this gene, or FP20, that confers this elongated fruit phenotype. Second is the fruit weight with tomato accessions that are showing increase in fruit weight are actually associated with a large 1.5 kb deletion in the fruit weight candidate 11.3. And last is the increase in locule number. Locule number are actually the compartment where the seeds reside within tomato fruits. And in this case, a large-scale chromosomal inversion resulted in a change in gene expression that facilitated the increase in number of the seed compartment within the fruit. So I've just shown that structural variation have actually fueled phenotypes that were actually relevant for plant biology, evolution, and agriculture. But nevertheless, the causative molecular mechanisms are not really well known and appear to be very diverse and largely uncharacterized. That's why it's particularly relevant to understand how structural variation can provide and diversify phenotypes that would be of agronomical interest. And by looking and understanding how structural variation works, we might be able to harness the structural variation to actually improve our current crops to make them more resilient for the fastly changing environment. So what's our approach? The idea here is to characterize the landscape of structural variation within our favorite crop model plant, which is tomato. So to do that, we undergone a very large sequencing effort by sequencing using long-read sequencing technology, 100 diverse tomato accessions. The idea here was to capture a large breadth of structural variation, to identify the structural variants and to classify them based on their feature, and then assess their consequences on phenotypes. So I'm showing here a phylogeny which is based on SNPs over 1,000 of tomato genomes. And what you can see here, following the different colors, are the different tomato taxons, with Solanum copersicum being the modern tomatoes, and then moving slowly towards early domesticates such as Solanum copersicum cerasiforme, and wild species such as Solanum chismanii, Solanum galapacensis, and Solanum tympanifolia. We sampled within the 1,000 of accession, 100 of tomato accession that were covering the entirety of the phylogeny in order to capture as many structural variants as possible. So the 100 tomato accessions selected contain 45 Solanum lycopersicum, 23 Solanum lycopersicum cerasiforme, early domesticates, and then 25 Solanum tympanifolia, 6 Solanum galapacensis, 5 Solanum chismanii that make the wild tomato species in our data set here. We sequenced them using the Oxford Nanopore long-read technology with mini-ion and mostly PromethION in order to have a huge quality and a high quantity of good data to look for structural variation. So then, using this data, we called the structural variation against our reference genome using sniffles and identified over 230,000 structural variants within the 100 genome. So we're providing here the first tomato pan-SV genome. And what the graph you can see here shows is actually showing the number of structural variants found per accession on the x-axis. So what you can see is that most of the accession we are looking at here are showing actually several thousands of structural variants. And structural variants, as you can see from the features on the right-hand side of the bar plot, actually being either deletions or insertions. Here, the fact that you see more structural variants in pimpinifolium and the wider relatives compared to the solanum-lycopersicum modern tomato on the right-hand side is because we call the SVs against the reference genome, which is a modern tomato. But eventually, there is a huge distribution of structural variants within the 100 tomato genomes we look at. We then wanted to know where the structural variants were found and what were the genomic features. So what we did and what we are showing here is the count of structural variants grouped for each tomatotype. So SP stands for Solanum pimpinifolium, SLC cerasiforme, Galapagos many are the wild tomatoes, and FLL is the modern Solanum lycopersicum. So what this shows is that structural variants are found largely distributed between various genomic features. And what you look more precisely at where those structural variants are found, we actually found that they are mostly, for a large proportion of them, found in the intergenic area, which is the pink box plot on the right-hand side of the graph. But a very large fraction are also found within the close vicinity of the gene. So either 5 kb upstream of genes, which is the green box plot, or within 5 kb downstream of the genes, which is the purple box plot. So this is particularly interesting because it means that there is a significant enrichment of structural variants in gene vicinity where regulatory sequence tend to cluster, suggesting that transposon might have, structural variants might have an important effect on gene activity. So we then wanted to know more about the genetic and the genomic features of the structural variants. And what we found is actually that most of the structural variants identified in our analysis were actually associated with transposable elements. 84% of deletions and 76% of insertion detected in our analysis are actually associated with repetitive sequence. This is huge. That is the main message we want to convey here, is that transposons are very major drivers of structural variation in the tomato genomes. What I'm showing on the stacked bar plot on the left-hand side is actually when you take these 84% of transposon-associated deletion and 76% of transposon-associated insertions, what you see is that at least 50% of these transposon-associated structural variants are actually due to what we call retrotransposons. Retrotransposons for the family Gypsy and Copia are transposable elements that have the ability to copy and paste themselves within the genome. And the fact that they make a significant fraction of the genome is here particularly interesting and suggests that the LCR retrotransposons from the Gypsy and Copia families are able to provide important variation within the genomes. I'm showing here a different representation of the same data, just to show you that when you are looking at structural variants that overlap with sequences annotated as transposable elements, you see that Copia and Gypsy retro elements are really enriched in the associated sequences, suggesting again that Copia and Gypsy LTR retrotransposons are major drivers of structural variation in tomato. So we then wanted to know more about the transposon landscape of tomato. And here we took advantage of our 100 tomato genomes, and we actually assembled 14 of them in order to have a more diverse idea of the transposon landscape within those genomes. And what we found, which was actually in line with what was known already from the current reference genome, is that at least 50% of the tomato genomes we have in our hands are actually made of transposons. And more than 90% of these transposable elements found in tomato genomes are LTR retrotransposons, either being Gypsy, which are the most enriched in LTR transposons in tomato, but also Copia elements that are also strongly influencing the transposon landscape in tomato. So what we show here is that structural variation is widespread in tomato. It is found in many different genomic features, with a strong fraction being found in promoters and downstream sequence surrounding genes, suggesting that they might have important role and effect on gene activity, and that transposons are major contributors to the structural variation, and especially Riderretrotransposon copy and gypsy. So because we saw that, we wanted to inquire more what was the biological relevance of transposons having an activity or consequences on gene action, on gene activity, and what could be the phenotypes that could be influenced by such activity. So that would be the topic I will be discussing in the second part of my talk right now. So I'm showing you here 4 different cases where phenotypic variation in tomato is happening. On the left-hand side of the slide, you can see huge variation in tomato fruit shape, and especially in this case, a very nice elongation phenotype that is happening in tomato fruit. On the top right-hand side of the slide, I'm showing you variation happening in the structure of the inflorescence, and particularly where the red star here denotes actually what we call joint. So the joint is actually a very important feature of tomato inflorescence because it allows fruit drop when the tomato fruit is ripe and mature. So in the J2 mutant, which is on the right-hand side, there's actually a mutation in the gene encoding J2, and it actually leads to the loss of the joint, which actually avoids the fruit to drop, which is a very important agronomical trait that has been selected by breeders for decades now. Bottom left, I'm showing you a very obvious change in fruit color with loss of function in the gene that is called yellow flesh, or R. And on the right-hand side, I'm showing you phenotypic variation in leaf shape in tomato. There is actually a common origin for such large variation within tomato phenotypes. In every single case, this is due to a change in gene activity due to a transposon insertion. And what is very interesting here is that for the 4 cases, a transposon from the same family is all the time responsible for the change in activity of the gene. And the transposon family involved here, and I'm just highlighting it now, is a transposon family called Rider. So we wanted to know more about Rider whereabouts and just to understand what made Rider such a strong driver of genetic mutation and such a strong driver of structural variation with potential strong effect on tomato phenotype. So what do we know about Rider? Well, Rider belongs, it's a LTR retrotransposon, so it belongs to the cochlear-like superfamily of transposons. And it's actually showing very classical structure with 2 long terminal repeats, LTRs, 5' and 3', that are flanking the coding region. So the coding region encodes all the proteins that are needed for the transposon life cycle. And what we know is that Rider is transcribed in modern tomato, but also in wild species related to tomato. So what you can see from the left-hand side is actually a northern blot showing Rider RNA. And on the right-hand side, you can see a blot showing actually obtained using probes that are actually detecting the long terminal repeats of Rider. So what you can see here is that tomato species and related species are showing Rider presence within their genomes, but then when you look at Solanum tuberosum, by example, which is potato, or then tobacco or coffee, then Rider sequences are not found within those genomes. So we wanted to know what was the distribution of Rider within the tomato genomes, and to do that, we used a sequence homology approach, and we de novo annotated intact rider copies within the tomato genome using our genome annotation tool, LTR-PRED. And we found 71 full-length rider copies within the tomato genome. As you can see from this figure, rider copies are found largely dispersed within the 12 tomato chromosomes, being found either in highly heterochromatic areas such as centromeres and pericentromeres, but also in euchromatin. The age of the transposon and the time at which the insertion event happened can give you an idea about the age of the transposon itself. So what I'm showing here as well, using the color code, is the estimated age of the transposition event, based on the similarity between the two LTR sequences flanking the protein sequence of the transposon, estimating the age of insertion. And what you can see here that we have a huge breadth of age of transposons, with some of them being very young, highlighted here in green, some of them being much older, highlighted here in red. So when we do a phylogenetic analysis on these 71 intact rider copies, we actually found a subpopulation of rider elements that are evolutionary young and then thus still very potent for transposition. And on the other side, which is about 2/3 of the rider population, rider elements that are heavily mutated that are showing low LTR identity within the sequences and suggesting that these copies lost their transcriptional and transpositional potential. We then wanted to know what was regulating rider activity. So to do that, we actually looked at the sequences that are contained within the long terminal repeats. So usually transposon promoters are contained within their long terminal repeats. And what we found through sequence analysis is that the LTR of rider actually harbors several cis-regulatory elements that are usually specific of dehydration responsive promoters. Cis-regulatory elements such as the MIPE-core, CBF, or ABA-related cis-regulatory elements actually markers of a potential activation by dehydration. So we tested this possibility, and through a combination of molecular genomic analysis, we actually found that environmental factors such as drought, water deprivation, and salt stress were actually able to activate rider transposition. And it was also interesting to note that alongside the environmental factors, so plant hormones, phytohormones, also have the ability to increase rider activity. On the other end, we also identify repressors of rider activity, because having an overly active transposon within genome can actually be pretty deleterious, right? And you're seeing a lot of genomic rearrangement. So we found out that within the tomato genome, it exists a surveillance system that maintains rider activity at a low level. And as has been found for the transposons, those repressors are mostly epigenetic, involving DNA methylation, small interfering RNA, and especially here in this case, in the case of rider, plant-specific RNA polymerase 4 and 5. Because rider is so active in tomato, we wanted to know if it was also found and potentially active in other related tomato species. And we looked at the presence of rider signatures, and signatures being the sequence of rider LTRs, within related tomato species such as Solanum arcanum, Penelei, and Abrocaetis. What this figure shows is that in arcanum, Penelei, and Abrocaetis, we've been able to detect very highly conserved LTR sequences belonging to rider in those genomes that are found here and shown here by this peak on the right-hand side of the graph that corresponds to very likely intact rider copies in those genomes. what these figures shows as well is that there is a huge number of remnants of rider sequences from these genomes that are shown here basically by the peak on the left-hand side of the figure. And what this says is that there has been strong rider activity in the past happening as well in these genomes. And these bursts of activity have been very well controlled by the genomes, and now we can see that the sequences are actually degrading pretty rapidly and losing homology when we compare them to the rider reference sequence. And we then extended our analysis further away in the plant kingdom and looked at the presence of rider sequences in other crop genomes. And what we found, surprisingly, was actually some really highly conserved rider sequence in a large number of crop plants, including here, by example, radish or wild strawberry, as well as beet or a carrot. So suggesting that the rider ancestor was already probably here in the last common ancestor before the split that led to this diversity of plants. And that also suggests that a rider or rider-like sequences have managed to stay active within those genomes, despite the presence of a surveillance system. So with this, I would like to put here my two take-home messages. So from the first part, I showed that the tomato pan-structural variant genomes actually reveal the extent of the structural variation we see in tomato species. And we also reveal the impact of those structural variants on the genome. And on the second part, was interested in transposons because we show that transposons are a main driver of structural variation in tomato. And transposons are very likely facilitated the diversification of phenotypes. Actually, transposons underlie a lot of phenotypes that are of importance for breeding and agriculture. So with this, I would like to wrap up and thanks, so first of all, my team and my supervisor, Zach, which is an amazing supervisor and gives me a lot of opportunity with this project, as well as Xingang Wang and Michael Lange, which are the main authors on the structural variant project. And I would also like to thank all our amazing collaborators that made all this research possible, as well as our funding, so HHMI, OYU's Medical Institute, as well as Cold Spring Harbor Laboratory. And with this, I would like to thank you for your attention. And I will be happy to take questions through email. Thank you.